Hey G Fun Facts Online fans, welcome back. Today, we're going on an adventure, not to a distant planet, but right under our feet. Seriously, we're diving deep into the dirt, the soil, to uncover a hidden world that could hold the key to the future of medicine. It's a biological frontier teeming with microscopic life, and it's absolutely mind-blowing. Imagine this. A single gram of soil, that's like a tiny pinch, can contain billions of microbial cells and thousands of different species. This isn't just random dirt. It's a treasure chest of what scientists call bioactive compounds. Think of these as nature's tiny chemical weapons, with the potential to become new antibiotics, powerful anti-cancer drugs, and other life-saving therapies. The field exploring this is called soil microbiomics, and it's exploding right now. But why the sudden urgency? Well, we're in a bit of a global pickle, especially with antibiotics. You've probably heard about antibiotic-resistant bacteria, or superbugs. They're a massive threat. In 2019 alone, antimicrobial resistance, or AMR, was directly responsible for 1.27 million deaths. That's more than HIV/AIDS or malaria in that year. And if we don't find new solutions, the economic damage could run into trillions of dollars by 2050. Now, soil microbes, especially a group called actinomycetes, have been our buddies in the past. They gave us awesome antibiotics like streptomycin way back in the 1940s. But by the 60s, the old ways of finding these drugs started giving us less and less, and new antibiotic discoveries dried up. So why the slowdown? Here's a huge roadblock. Most of these soil microbes are incredibly picky. They're like divas. It turns out, only about 1% of bacteria from the environment can be grown in a standard lab petri dish. That means a staggering 99%, about what scientists call the unculturable majority, have been out of reach, along with all their potentially amazing medical compounds. They just don't like our artificial lab setups. They have very specific needs that we haven't been able to replicate. But science, as always, loves a good challenge. Researchers are now using some incredibly clever new tools to tap into this hidden pharmacy. First up, there's metagenomics. This is super cool. Instead of trying to grow each microbe, scientists just suck out all the DNA directly from a soil sample. All of it. Then they sequence this collective DNA. It's like getting the blueprints for every single organism in that community. They can then hunt for specific genes or clusters of genes, known as biosynthetic gene clusters, or BGCs, that are responsible for making these valuable compounds. This approach has already led to discoveries like melacidins, a new class of antibiotics that can fight multi-drug-resistant bacteria. Then there are novel culturing techniques, especially in situ cultivation. Scientists figured, if microbes love their natural home, let's try to grow them there, but in a controlled way. Enter the iChip. This nifty device is like a tiny microbial hotel. It isolates single bacterial cells from soil into tiny channels in a diffusion chamber. This chamber, covered by special membranes, is then placed meter back into the soil. Nutrients and growth factors from the natural environment diffuse in, helping these previously unculturable microbes to grow. And guess what? One of the superstars discovered this way is teisobactin, a promising antibiotic that bacteria haven't shown resistance to so far. How amazing is that? Next, we have genome mining and synthetic biology. Even for the microbes we can grow, sequencing their DNA has shown they have way more potential to make cool stuff than we thought. Many of these BGCs are silent in the lab, meaning they're not switched on. Genome mining helps find these silent treasure troves. Then, synthetic biology tools, like CRISPR gene editing, let scientists wake up these silent pathways, boost production of the good stuff, or even design completely new molecules. It's like being a molecular DJ, remixing microbial genes for medicine. And finally, helping us make sense of all this incredible data is artificial intelligence AI and machine learning ML. Metagenomics and high-throughput screening generate absolutely massive amounts of information. AI and ML algorithms are like super smart assistants that can sift through this data, spot patterns, classify microbes, predict if a compound will work, and even forecast antibiotic resistance. They're massively speeding up the whole discovery process. And it's not just antibiotics. Soil has already given us a pharmacy's worth of amazing drugs. The anti-cancer drug doxorubicin, sometimes called the Red Devil, came from a soil bacterium found near an old Italian castle. Bleomycin, another anti-cancer agent, also has soil origins. The immunosuppressant cyclosporin, vital for organ transplant patients, and the cholesterol-lowering drug lovastatin? Yep, soil microbes. And let's not forget ivermectin, a true wonder drug against parasites, discovered from a Japanese soil sample. With all these new technologies, the hunt is back on. Researchers are exploring soils from all over the world, remote forests, deserts, even unique spots like caves and hot springs. Peat soil, which is super rich in organic matter and microbes, is looking especially promising as a largely untapped source for new antibiotics. Now, it's not all smooth sailing. Soil microbial communities are incredibly complex, and we still have a lot to learn about how they work. Finding and making these new compounds, especially from the unculturable ones, is still tough. And the sheer amount of data from all these omics techniques, genomics, proteomics, and so on, needs some serious brain power and computing power to analyze. And then there are the ethical questions. When we find these amazing things in nature, who benefits? It's crucial that bioprospecting, this search for nature's treasures, is done responsibly. This means sharing benefits fairly with the countries and communities where the samples come from, something outlined in agreements like the Nagoya Protocol. We also need to balance protecting intellectual property with open science and collaboration. And if we start engineering microbes, we need to be super careful about any potential impact if they got into the environment. So what's the takeaway? Prospecting Earth's microbial diversity for new medicines is a huge, complex job, but the rewards could be phenomenal. It needs experts from all sorts of fields working together: microbiologists, chemists, geneticists, computer scientists, ecologists, you name it. International teamwork is key, and even citizen science projects, where you folks could contribute soil samples from interesting places, can help broaden the search. The future of medicine might truly be hidden right under our noses, or rather, our feet, within that amazing web of life in the soil. By keep innovating, exploring, and working together responsibly, we can unlock the secrets of this microbial treasure trove. This could lead to a whole new era of medical breakthroughs to tackle some of the biggest health challenges we face. It really makes you think differently about a handful of dirt, doesn't it? The rich biodiversity of our planet's soils holds so many keys, and soil microbiomics is the science that's helping us turn them. All right, that's all the fascinating facts we've dug up for you today on Fun Facts Online. What did you find most surprising? Let us know in the comments below. Don't forget to like, subscribe, and hit that notification bell so you don't miss our next deep dive. Thanks for listening, and we'll catch you in the next one.